Welcome everyone. First let me introduce what will be next week. Next week we will have a talk again in Czech. It will be Klára Holková from Department of Cell Biology, and her title is a coincidence or necessity, and it will be about why we use just these 20 amino acids in our protein. So it will be another nice talk to come in a week. Today let me introduce Roel Wouters, who joined our team this year, and Roel will probably introduce himself.
Very briefly.
Yeah. Very briefly, it's enough to have more time on fascinating topic about coevolution of snake venoms and victims. So hopefully you are not that scared and stay alive.
Thank you, Lukáš, for introducing me, and thank you for inviting me to the whole organization. Also, thank you for enabling me to do it in English, because my Czech is terrible, so this would be a true disaster. Stayin Alive, for coevolutionary warfare. I hope at the end of this talk you'll be interested as much as me in venom evolution, and especially why I think it's a great model to study evolution. First of all, these three guys are the Bee Gees. Well, the organizer asked me to come up with a funky title. I generally don't like funky titles for scientific talks. But okay, inspired by my dad, who is a real Bee Gees fan, I decided to go with Stayin Alive. And last week I was at the first aid course that Lucia organized with her team from the doctoral office. And of course you need to do CPR on 100 beats per minute, so we used the Stayin Alive. And then I found out last week there's a Czech version of it. Hoda. So if you don't know it, now it's in my playlist, so if you don't know it, go and listen to it. It's from 1976, but it's exactly the same beat, exactly the same melody, but in Czech. All right, let's get started. So, as Lukáš introduced, I'm now a PhD student with Lukáš group, together with Lukáš, Maria Admanova, at Charles University here, but also at Czech Academy of Science in Liběchov. But in my previous life, being my bachelor's and master's, I studied at Leiden University in the Netherlands, where I'm originally from. And I was not born, but I was definitely raised in the group of evolutionary and developmental zoology by Professor Dr. Michael Richardson, the professor over here. He's an absolute legend in embryology of birds and reptiles. And at the time, they had a lot of snake venom projects going on related to evolution, ecology of snake venom and venomous snakes. And I joined the lab there in 2018 as a bachelor student, and I kind of never left. I did different projects as well, but I always stayed attached to the group and did a lot of side work in the group. And several papers came out of this few years that I worked there. And I will today touch upon these three, two of them covering toxin resistance evolution, and the last one covering our work on spitting cobra venom. So let's get started. Oh yeah, I always have to mention Yori. Yori and I joined the lab in 2018. He became one of my best friends. We did all the work together, so whatever I'm saying, he can say too, and whenever I'm presenting, he could tell you exactly the same story. So always think of us together when I'm giving this talk. He's now a PhD student with Sean B. Carroll in Washington, D.C., so that's why he's not here. So today's talk will be a little bit in the framework of convergent evolution. I'm quite sure you all know what is convergent evolution, but just to put it in perspective, convergent evolution is the independent evolution of similar features in species of different periods of time. And my favorite example in vertebrates is, one of my favorite examples, you will soon know a few more, are wing evolution. As you can see, in 3 clades, well, extinct in reptiles, but birds, bats, you know them. They did something weird with their forelimbs, and now they can fly. But they are distantly related from each other. They don't have an ancestor with wings. So this is what we call convergent evolution. And now I'll show you a little video to get in the mood of the talk today. So there's no sound, so I'll talk you a little bit through it. But it's quite an iconic and often filmed match between mongoose and cobra. This one was filmed by Smithsonian, but National Geographic has coverage of this as well. And they have a predator-prey relationship, in this case the mongoose being the predator. And here you can see the mongoose receives a full bite of the venomous cobra, but the mongoose runs off, and he is fine, he survives the bite. And we have known since the 90s that they have some sort of resistance against the venom of the cobra. So today I will go more into depth on how this works, but you have to see it in the light of a kind of an evolutionary warfare, millions of years of trophic interactions that lead to toxin resistance. So the predator or prey need to become resistant to overcome the effects of the venom of the snake, or in this case the toxic newt and the garter snake, which is a very well studied example. So it's kind of an arena where resistance and poison or venom or toxins in general are intertwined in a very long-lasting millions of years of co-evolutionary arms race. And if you look at toxin evolution in general, over the entire animal phylogeny there are millions of toxins. I'm not even talking about plants or bacteria or whatnot. I'm only talking about animals here. And we know that there are over 200,000 toxic species of animals, and we know that it has at least 101 times independently evolved within the animal phylogeny. So this is also, well, we think of venom and toxin poison as something special, but as a matter of fact, it's quite common. I like to see venom in snakes as a functional trait because it kind of mediates antagonistic interactions. And in snakes, they use it for prey acquisition, and as we will see later, they also use it as a defense, in the case of spitting cobras. What you have to understand throughout this talk is that if I talk about venom, I mean a complex mixture of many toxins, tens to hundreds of toxins, and I like to think of it as a cocktail, just like this Bekherov Camille. Although Bekherov Camille consists only of a few ingredients, venom can consist of many different components. And just for your information, the difference between poison and venom, very often it's used as, if I bite it, I will die, it's poisonous. If it bites me, it's venomous. But I like to think of it in a bit more sophisticated way, is that poisons almost exclusively consist of small molecules of alkaloid or steroid-based low molecular weight molecules, while venoms are almost exclusively made of proteins, and very often a combination of many of them. So we wrote a large review on venom, toxin, and poison resistance. I use these three terms a bit through each other, but I will stick to toxin resistance as it makes more sense. It covers both poisons and venoms. And we described basically 3 types of resistance. Very common is predator resistance, where a predator has evolved some resistance against a toxic prey. In the case of mongoose, the cobra. In the case of the garter snake, the tariga. nude. On the other hand, you have prey resistance, where the prey that is being eaten by the toxic animal evolves some kind of resistance. And also more and more described is auto-resistance, wherein a snake, or in this case a cobra, as we find it in cobras as well, but many animals have some form of resistance against their own toxins, which might be very useful. So in this case, they are substitutions, so small DNA mutations that mediate resistance. Often small changes in the makeup of genetic receptors that play some sort of function in the physiological processes. We also describe some of the inhibitors that are in our circulatory system and kind of use as scavenging, almost like an antibody kind of a mode of action. I will not discuss them further. There are just a few of them, and I will focus on the examples of substitutions on the receptor level. One example I would like to give is cardioglycosides. They are a poison, a toxins, group of toxins found in many animals, including toads and also rhabdophis snakes, for example, the poisonous snakes. And they are steroidal compounds. And what they do is they inhibit the sodium-potassium pump. And because of that, sodium outflow is blocked, calcium increases, and it results in cardiac contractions with greater force, cardiac dysrhythmia, and tachycardia. And for many prey species, it means the end of you. However, there are many animals, including predators and some prey, that evolved small substitutions in the extracellular domain of the sodium-potassium channel that resulted in a positively charged binding domain, which causes electrostatic repulsion because cardioglycosides are positively charged. So that's how they bounce off, functionally bounce off the toxins. And we find it all over the animal kingdom. We find it in, for example, our European hedgehogs. We know that they eat toads, and they can actually deal with the poison in the toads. We find it in many snakes, including all the naias. We find it in rhabdophis, obviously, as they eat toads, but they also use the poisons to store in their own poison glands. We also find it in monarch butterflies that themselves are also poisonous, having these cardioglycosides. And a special case I want to talk about is the monitor lizard, and that's also why I like this example. Because we see that the monitor lizards from Africa and Asia indeed have these substitutions and are therefore resistant against cardioglycosides. But the Australian monitor lizards, we did not find, and also literature did not find, any resistance against these cardioglycosides. And we know this with the introduction of the giant toad, the cane toad, in Australia for pest control. It was also the first introduction of cardioglycosides to Australia. There never before had there been cardioglycosides in animals in Australia. They eat the poisonous toads, and they suffer from the consequence of the poison. So this is actually how this kind of molecular framework has actually quite some impact on invasive species. Another example which I really like, and then we go to the snakes, I promise you, are those of pain-inducing scorpion toxins that we found in bark scorpion Centauroides. So Centauroides is a large genus of scorpions that are found in North Central South America. And they have pain-inducing toxins that activate voltage-gated sodium channels, specifically 1.7. And there is one mouse, it's the only described example, one mouse, it's the grasshopper mouse from the United States, that really likes to eat these bark scorpions. And they have substitutions in the sodium channel 1.8. And actually it attracts the positively charged toxin, and the toxin binds to 1.8 instead of 1.7. And to me that's quite interesting, because this is an example of off-target repurposing, which is for toxin resistance very rare, wherein a different sodium channel kind of attracts all the attention for the toxin binds to that place, but it doesn't have any function in the 1.8. So it doesn't induce pain in 1.8, it does in 1.7. So this is I think a genius way to solve your problems of target repurposing. It's just a small example, it's only described in this mouse, but I like it, so that's why I put it in there. So now we'll go to the snakes, and we are going to talk about snake venom alpha neurotoxins, because they are by far the best studied group of toxins when we talk about venom resistance. So alpha neurotoxins, they belong to the so-called three-finger toxin protein complex. It doesn't really matter, but here you can see three-finger toxin, and it kind of has three elongated parts in the structure, so that's why they call it three-finger toxin, but that doesn't really matter. Just what is important to understand is that they are the main component of elapid venoms, elapid being cobras, coral snakes. They can be either neurotoxic, as we will discuss now. They can also be cytotoxic, which we will discuss later. But they are an important group of proteins in snake venom, especially in that of elapids. And what they do is they bind to the postsynaptic nicotinic acetylcholine receptor. So back to high school, here we see the synaptic cleft or synapse gap. And this in red is the quite important neurotransmitter acetylcholine, which plays an important role in involuntary muscle movement, especially from neurons to skeletal muscle. And they are the most important neurotransmitter in the parasympathetic nervous system. And normally they bind to these postsynaptic nicotinic acetylcholine receptors, but alpha neurotoxin does the same. It binds to these postsynaptic nicotinic acetylcholine receptors. So it's an antagonist. It blocks the normal neurotransmitter from binding, and it leads to paralysis and immobilization. And this is the primary mode of action of prey capture in elapids. So if you look at the structure of this postsynaptic nicotinic acetylcholine receptor, we see it consists of some subunits, and very conveniently, alpha neurotoxins, just like acetylcholine and some conotoxins that you can find in cone snails, bind to the alpha-1 subunit. So alpha, that's easy. And this is the same picture, but then in a tertiary structure from our paper. Here you can see the alpha-1 subunit in green, the ligand binding domain, and the three-finger toxin attached to this subunit. So, both literature and I think we added about 148 new species of predator and prey all over the vertebrate kingdom show some sort of substitutions in the ligand binding domain of this subunit. There are different mechanisms, I will go into them in a second, but they have pretty much the same function. They prevent or disturb the binding of alpha neurotoxin. So a little bit on the mechanism. These are the sites in the ligand binding domain, and these can be the mutations you will find, substitutions. And one example is an N-glycosylation event, which causes steric hindrance, and as you can see depicted here, it forms some sort of, well, kind of a blockade or at least some disturbance, and it means the three-finger toxin cannot bind, but the acetylcholine can bind. Another example is electrostatic repulsion. I already mentioned it, but what happens is that you get either an arginine or a serine replacement, and they are positively charged, resulting in a positively charged binding domain repulsing the three-finger toxin that's also positively charged. Luckily, acetylcholine can still bind. And the other form we find is a proline substitution into, they can be lysine or histidine, but they can actually be other forms as well. And they probably have some different kind of conformation, but it's not really known what happens there. We call it mechanical, but we don't really know. But these are the three types of substitutions, or at least the effects of the substitutions we find when we're talking about alpha-neurotoxin resistance. So just some animals that were already known or we described that have these alpha-neurotoxin resistance. So, for example, honey badger, they have glycosylation. They have electrostatic repulsion, so positively charged substitution, and we know they eat everything. They are also resistant against those cardiac glycosides we discussed before. They are, well, sometimes they say honey badgers don't give a fuck. They eat everything, and they are not to be killed. We find it also in African reedfish. A bit weird, maybe. We know from some literature they are being eaten by Naia annulata, which is a fishing cobra. But we also know that there are some algae that produce similar alpha-neurotoxins, so maybe it's because of algae in the water. But they actually have two different substitutions causing both steric hindrance and also this electrostatic repulsion. We were the first ones to describe it in the very well-known meerkat that you know from the zoo or from the wild if you've seen it. They have both steric hindrance and also this proline substitution, resulting in some mechanical resistance. I will get to talk about caecilians in a second. Cobras, actually all elapids, have some mechanical and some steric hindrance substitutions. This is maybe difficult to see, but this is a Bitis arietans, a puff adder. So also vipers, many vipers, not all of them, but many vipers have resistant motifs. Also mechanical, but a different substitution compared to cobras. So it seems like they have conversely evolved. And they also have some steric hindrance. Okay, and then very famous, everyone has had it as a kid, or at least some of the biology students here, I reckon. Pogona viticeps, it's known to eat elapids with Australia's full of, and it's also known that elapids like to eat pogonas. So they also have similar substitutions as the meerkat and the cobra, and they also have steric hindrance. So this is just some examples. We found many, many more, but just some examples from the phylogeny. And one specifically I want to point out is a recent publication from last year from our colleagues in Australia that looked at resistance in specifically one lineage, the lineage of caecilians. And for some reason, caecilians, many species, display some motifs of actually all the different kinds of substitutions that are possible. They even described some new substitution that confers resistance. And interestingly enough, and this is why I wanted to point this out, there is one group of caecilians that are on the Seychelles that do not have these motifs, as they are also allopatric with elapids. There are no elapids on the Seychelles. They are not exposed to these toxins, and they either never evolved it or they lost it. But this is, yeah, a quite interesting gap. While most of the species that they looked at from the Africas, from Asia, from the Americas, they all show it. And it's known, this was published actually last week. I don't know if you've seen it, but it's known that elapids like to eat caecilians. And this was a kind of weird example of two micrures, two coral snakes eating one caecilian. And one lost and one won. Yeah, you can see there's a movie. If you check the movie, I didn't put it in, but if you check the movie, you can see the caecilian actually lives all the way until it's being devoured. So it doesn't suffer from the effects of the neurotoxin. Okay. However, we do find some constraints to resistance. So here you can see the same ligand binding domain again from that alpha-1 subunit, but now just in amino acid sequence. And you can see the larger the letters, the more conserved this region is over the phylogeny. So you can see there are some vital amino acids that apparently you cannot change too much, or you will lose receptor function. And there are some places that are more prone to mutations. And we know that toxin target vital physiological processes. Receptors are essential, and that's probably why they are highly conserved regions. And there are limited possibility for mutations to occur on this ligand binding domain, and constraints on sequence plasticity while remaining this incredibly important receptor functionality. It's really crucial for neurotransmission, for signal transduction. And that's why you see this, well, similar substitutions all over the animal kingdom. Okay, there's one case that I wanted to highlight, maybe give you some food for discussion later. We don't really know, but European vipers from the genus Vipera, we don't find any substitutions that confer resistance, but yeah, they either have some sort of reversal, or they never evolved, resistance in the first place. But we know that they are allopatrically lepid since the late Miocene, and maybe because of the selection pressure, they might have lost it. But we don't really know. So we discuss it, but we don't know what's the reason. Okay. So then a little bit about birds. Is molecular resistance the only way to survive? Well, we did not find any genetic resistance in birds against alpha neurotoxins. Recently, people looked at SVMPs, which are rattlesnake toxins. They did not find it there either. So far, there has been no molecular resistance described in birds to any toxin. And we kind of came up with the idea that maybe birds have existing morphological traits and some specialized behavior to negate the selection pressure for molecular resistance. So we took the secretary bird as the main example, but you can translate this to more snake specialists. They have often long legs, not always, but often long legs, which have minimal vascular tissue. So if you get a bite in there, it probably will not affect you that much. They can fly. Obviously, they have very good eyesight. They're agile. They have often very specialized behavior to kill snakes, like secretary birds use their long feet. Crested serimas from South America, they kill the snake by throwing the head on a stone. We know also in these giant hornbills they do the same. Snake eagles often attack by ambush, getting the snake behind the head, killing it immediately by the claws. So in general, the point is that many animals don't always rely solely on genetic molecular adaptations, but they also have some behavioral or morphological adaptations And one example you can find in ground squirrels. Ground squirrels have resistance against snake venom metalloproteases in rattlesnake venom. However, on top of them being resistant against this toxin, they still have some special trait where they use the tail wiggling to attract the attention of the snake. And interestingly, when you compare the rattlesnake to a non-venomous gopher snake, which is sympatric, rattlesnakes are pit vipers and they can sense heat. They have infrared sensors, they can sense heat. And it sends a lot of body warmth to the tail, so it actually warms up its tail, wiggles its tail to distract the rattlesnake from biting in the tail. Most likely, being bitten in the tail, far away from the vital organs, increases your chances of surviving it. Because I have to address, I'm constantly talking about specific toxin resistance, but obviously venom is a complex mixture, so it doesn't mean that if the snake bites a squirrel, it has some resistance against some toxins, but there might still be toxins that actually affect it. Maybe not kill it, but still have some detrimental effects, causing some sickness or some local injury. So being bitten in the tail is probably better than being bitten in your vital organs. There is a beautiful website called ninjared.org if you want to see more of this cool footage. Also in other species, go there. There is a whole array of adaptations, behavioral adaptations, that rodents evolved to negate bites from snakes. So it's not all. There are many more toxins in the animal kingdom. And so there are also many more genetic patterns of resistance. We discussed a few, but the review is very big, so I will not go into all examples. But it is widespread among toxins all over the animal phylogeny, and among animals all over the animal phylogeny. So very often I got the question. So this would be normally the end of my talk, and very often I got the question like, but can we use this information to, CRISPR-Cas it in humans and make us survive snakebite? And I always said it's maybe some food for thought for the future, but actually just a few weeks ago, 21st of February, a very interesting paper came out where they show in a in vivo mouse model, where they actually put the receptor, the binding domain of the receptor in an antibody, and they show that the mouse that has the antibodies survives the effects of alpha-neurotoxin injection. So now this information is put in a model. Who knows, it might go through some, more advanced stage in the future. So what about resistance in humans in general? This is that's a paper, yeah. The paper by Richard Harris and colleagues from Australia. They looked at binding affinity. It's in vitro model system. So they kind of put the receptors of these primates and the alpha-neurotoxin together, and they looked what would happen to the binding affinity, and what they find is that in lemurs and in South American monkeys, there is a very high binding affinity of alpha-neurotoxins, while in old world monkeys, and especially in apes, great apes, including ourselves, they show a very low binding affinity, suggesting that there might be some mechanism of resistance in place, but there is no genetic basis for it. So we don't find any substitutions that kind of confer resistance. There is only this paper, but it kind of shows like, okay, maybe on Madagascar there are no alpha-neurotoxic snakes, so they never needed to evolve some sort of anti-susceptibility. Central South American monkeys, They might deal with micrures, but micrures, they are often fossorial snakes living on the ground, and monkeys often in a tree, so maybe they do not really deal with elapids. And we know old world monkeys, they are sympatric cobras, and also well known to engage with cobras, so they might have some. But in general, it's quite interesting to look at humans and snakes, and this is maybe, I watched back in Czech, but I watched back your talk, Daniel, from a few weeks ago, so there might be a bit of overlap, but humans and snakes have quite been involved in a co-evolutionary arms race themselves. And this is a video I want to show you, and I want to give you a task with this video. Try and see at which step you can identify the object. Oh, wait, I have to now do it again, sorry. Well, we can do it once more. If it works. There we go. It ends with a graph, and I need the graph, so we have to do it again. Once more, practice. There it is. So as you can see, and maybe intuitively already realized, is that humans are able to detect snakes faster than other animals, including animals we deal with every day. Most of them could already, before half of the picture was clear, could already see the snake. This led, well, part of this work led to the snake detection theory or hypothesis as proposed by Lynn Isbell. And she did fascinating work on macaques, where she showed that the response magnitude and response time actually towards pictures of snakes in macaques is, well, significantly higher than to parts of the same species or some random objects. And it led to the formation of the snake detection theory, and it's proposed that the superior colliculus, which is a part in the midbrain that combines vision to movement to space, it sends it to the pulvinar, which is a visual attention center, and then sends to the amygdala, which actually puts emotion to what you see and is able to infer a fear-related stimuli, which works as a reflex. And we see that for snakes it works at a low spatial frequency. It works even at very, very quickly, as you can see in the next picture. So she found like 91 neurons are activated in this pulvinar. And this is unique to primates, especially dorsolateral and medial. There is no homologous structure in any non-primate mammal. So it seems to be a unique trait to primates that both in time, magnitude, and proportion, they can distinguish snakes. This is the same work on macaques. They can distinguish snakes very quickly. This is just a matter of milliseconds, like a reflex from... In the beginning, they can already see the snakes. Then later, they can distinguish the snakes from anything else. And then in the last stage, they can distinguish all the different objects. But snakes always stand out immediately. So it kind of shows that we have a special detection center for snakes. And I think it kind of relates to modern day history, including the Bible, where Adam and Eve are seduced by the snake. or ancient Egypt. These are snake mummies we have in the Leiden University collection, and these were micro-CT'd, and you can see that Egyptians often put gravid snakes. Mummified gravid snakes is a sign of fertility. In Hindu culture, religion, it is also sometimes a sign of wisdom and fertility. We all know Samuel L. Jackson's Snake on a Plane. I think it's his best work ever. Really working on this fear of snakes. Slytherin, Harry Potter fans. It all boils down to this kind of snake detection center and this fear of snakes that we have. And it kind of makes sense that we have it, as currently snakebite is the most deadly tropical neglected disease out there, causing many casualties every year. It was late Kofi Annan, former Secretary General of the United Nations, who kind of advocated for snakebite and to put it on the international agenda. He called it the biggest public health crisis you've never heard of. And many villages, almost every community, village, city in rural Africa, parts of Asia, parts of South America, they deal with snakes on a daily basis, and they deal with the effects of snake bites every day. And it probably has been this way for millennia, maybe millions of years. So there's probably a lot of reason to be afraid of snakes. So we know snakes have kind of left their evolutionary marks in us, but did we also influence snakes in any ways? And now I want to introduce you to the last part of the talk, which is spitting cobras. So first a short movie. You can see a plastic screen with a kind of a face on it. Here you can see a Naya pallida, red spitting cobra, and Hemagatus, the rinkhals. It's not a real cobra, but very close relative to the cobras from southern Africa. There you saw Professor Nick Casewell from Liverpool School of Tropical Medicine, with whom we collaborated. And this is actually the way we collected the venom for our studies. So we would let them spit on a fake face, scrape off the venom let the venom dry, scrape it off, and use it for our essays. So this led in the end to our publication in Science magazine, and this is what I will discuss now. So spitting cobras, there are 3 clades of spitting cobras, 16 species, 8 African species that are the Afronaya clade, 6 Asian species, and it used to be 1 rinkhals, but actually a few months ago there was a second species of rinkhals described from museum material, might be already extinct, might have some ruminant populations, we don't know yet, but now I will have, for the completeness of it, I will say there are 2 rinkhals, but they are monotypic genus, so it's just 1, or now 2 species in the genus, close relatives to the cobras. And they use it for prey capture, just like any other cobra, and they use it for defense, so for deterring some opponent. And they have special adaptation, the fangs. So here you can see the fangs of normal non-spitting cobra, and here you can see the fangs of a spitting cobra, and you can see that they have kind of smallened the gap and made it a bit more oval, so they can actually spray the venom instead of inject it like a hypodermic needle. And it's very effective as long-distance defense, so it's extremely accurate over 1.5 to 2 meters. Yep. And this is what it does to your eye. It causes all kind of irritation, pain, coagulation, and if not treated, and treatment is quite easy, you just have to wash it out of your eyes. But if it's not treated, it can ultimately lead to blindness. And it sounds easy enough, putting water in your eyes, but just imagine you are in rural Africa, you are walking to the water well to get your water, and you are being spat in the eye by a red-spitting cobra. It's not that automatic. You always are in close distance to water. So there is Jori again. Part of his study he did in Thailand, where he worked on king cobras. They don't spit. They don't spit. But in the time he was in the field, he was spat in the eye by this beautiful Indo-Chinese spitting cobra, Naia siemensis. He went to the hospital. They washed it out. It was all fine. Nothing. No big deal. But he came back to Leiden, and he said, "Okay, Roel, just this happened to me. What effect does it actually have on your eyes? I want to know everything about this venom now. I want to know what does it do to your eyes and how does it work?" So together with Professor Richardson, we kind of reinvented a model for eye irritation potential, which is called the Hans-X test, the chorial allantoic membrane, which is this outer membrane of the chick embryo. And the setup was very easy. We put spitting venom in different concentrations, non-spitting venom in different concentrations, some controls like the casein from red peppers, for example. And what happens over time? Yeah, I have to say this model, I put here L'Oréal Paris, but this model is still used nowadays in labs of many shampoo, perfume developers that still use this test as a potential of eye irritation. So it's not, it was invented in the 80s, but it's still used nowadays. We repurposed it for a different test. So basically we took the venom, removed the eggshell, put it on the membrane, and see what the effect is. So we looked at all of these species, some spitting cobras, some non-spitting cobras from both Africa and Asia, rinkhals, some outgroups of related elapids that do not spit, Africa, Asia. And I just want to show you the most extreme example. This is just one example. I will show you results in a second, but just one example. Here you can see Cape cobra, non-spitting cobra, and here you can see Mozambique spitting cobra, spitting cobra. So just this is a time lapse of 5 minutes. Just enjoy the show. So there seem to be clear differences between spitting and non-spitting in this example. In this case, you will see some hyperemia, you will see some coagulation, and if you wait long enough, even after this 5 minutes, some of the coagulated veins will burst, and you even get hemorrhage. So these are the results for all the species we tested and all the replicates we did. And here you can see this is the irritation scale, so to say. You can see in the outgroups and the African non-spitting cobras, absolutely no irritation. In the African spitting cobras and in Asian spitting cobras, and in some extent also the Asian non-spitting cobras, they show all kind of irritation. So it doesn't explain the entire story, but at least it gives some indication of these differences. So another test was needed, and then the colleagues from Australia, they did some sensory neuron activation assay, where they took some sensory neurons from the trigeminal, which this optic branch, which also innervates part of the eye, and they looked at the cell activation as a measure of pain. So now we have irritation, and now we look at pain. And they see in the outgroups, in the African non-spitting cobras, absolutely no activation of these pain receptors. But if you look at the spitting cobras, rinkhals, spitting cobras, and these are the non-spitting cobras to some extent, some of them a little less, some of them a little more, they show distinct activation of these sensory neurons, trigeminal nerve. So then in Liverpool, the leader, like the group leaders of the project, they looked at the venom composition. And again, I want you to remember, venom is like a cocktail, so it consists of a lot of different toxins. And here only displayed the main components. So as I've told you before, in blue you can see these are all the three-finger toxins that we talked about. In dark blue they are the neurotoxins, and then in light blue they are the so-called cytotoxins that have some disruptive effect in the cells. And then what we strikingly see in these spitting clades is they all have a higher proportion of the so-called PLA2s, which are phospholipases that disrupt the cell membrane. And we see this is typical for all these spitting cobras. And if you look at the cell activation of the specific toxins within the venom, so not the entire venom, but just the specific toxins, you can see that in itself, PLA2s, they don't cause pain. Okay, cytotoxins, a little bit. They probably enter the cell a little bit and do their disruptive work. But then the combination of these cell membrane disruptors and the cell disruptors, they cause this spike in activation of these sensory neurons. So our conclusion is that the upregulation of these phospholipases are in synergy with already present three-finger toxins, and they cause extra pain and irritation. And that's the first time that we describe a synergy, a collaboration, as it may, between two toxins within the same venom. So on itself, not much effect. In combination, a lot of effect. So then, a few years ago, an Australian lab already described the correlations between cytotoxicity, so this three-finger cytotoxicity, hooding behavior in snakes, so these cobras that have this hooding behavior, and spitting behavior. Actually, all of these cobras, even the non-spitting ones, have to some extent PLA2s in their venom. So they basically just upregulated some of the pre-existing PLA2s to increase the proportion of that in their venom composition. We know that cobras already have this hooding behavior. They already have this kind of behavior towards predators, opponents, that they would elevate their posture. So they're already in a kind of a position where spitting might be effective. And they have some fang adaptations which are basically not much more than smaller opening. So now we know they spit and we know how it works in their venom, but why did they start spitting? And this is where it gets interesting. Food for discussion. But we believe that our own ancestors might have played a role in this behavior. So about 6 million years ago, we started to walk on two legs. We diverged from the other apes. And about 4 million later, about 2 million years ago, we first emerged into new areas, including Asia. And this is the same graph, but then different. And now I point the estimated times in which cobras started spitting. And we see that in Africa, they started spitting roughly 6 million years ago. And some 4 million years later, about 2 million years ago, the Asian cobra started spitting. These are relatively recent innovations in 40 million years of cobra evolution. And we know that apes use tools, not only apes, even other primates use tools, including stones and rocks. And in the case of hominins, they carve weapons, they use weapons even to deter and kill snakes. And we know that many predators, including mongoose, birds, and whatnot, they use close combat strategies. And spitting is effective over a large distance. It's kind of a long distance weapon. And there is a huge lack of data on observations of spitting behavior against predators like birds or other predators like mongoose. And then there is the alternative hypothesis that they might spit because of ungulates that are large ungulates that are on the savanna systems. But they have their eyes sidewards, and we know that these spitting cobras are very good in spitting directly in our eyes, especially in those of our front-faced predatory eyes. Even when you change the conformation of the head a little bit, they always know where to spit. And this study also shows that they spit more often at faces than at different objects like hands. So it seems to be some cue, a human-like face, an ape-like face that kind of triggers this spitting behavior. So there was an illustrator after the science publication came out, Nundu. He made this beautiful picture of one of the first two-legged hominins on the savanna, Kilimanjaro in the back, and then a beautiful spitting cobra spitting towards the eyes of the early human. And I like to think that we are part of nature and have shaped evolution in our own way, human-wildlife interactions in a super ancient context. So with that, I wanted to end. Million years of trophic interactions lead to the emergence of toxin resistance all over the animal kingdom. Constrained by vital receptor functioning, remember the case study of the neurotoxins. Humans and snakes seem to be intertwined in a coevolutionary arms race. We have specific snake detection center, our pulvinar, which is unique to primates. We have 3 lineages of elapids that have evolved their venom for spitting behavior, causing immediate irritation and pain to the eyes. And we propose that two-legged hominins and Homo erectus are proposed drivers for spitting adaptations in both these African and Asian clades. Snake venom evolution and toxin resistance are good models to study. Convergent evolution, I think, nice models to study evolution in general. Especially thanks to Yori, Professor Rich, and Nick and Wolfgang, and Muzaffar, and Sven for the beautiful illustrations of our paper, including the one of the secretary bird. And with that, I want to end. Thank you.